In this video, let us study about what is rDNA and the steps involved in rDNA techniques. It is one of the important topic. Let us know what is rDNA. R means here recombination. Recombination DNA is the hybrid combination of two DNA fragments which is derived from different sources. Here it is a hybrid combination of two DNA fragments which is derived from different sources. This definition you will be clearly understand when you know about the steps involved in rDNA techniques. Okay, then you come to know about the meaning of this definition. Okay, let us study about the steps involved in recombinant DNA. Okay, here we already in the definition we studied that we have derived from different sources, DNA fragments which is derived from different sources. Yes, let us take the two different sources, okay, containing DNA. The first is the bacterial cell. The bacteria is E. coli bacillus, and the another source is the human cell or plant cell, whatever it may be. And the speciality of the bacterial cell, we already know that the plasmid present in the bacterial cell. In this technique, the plasmid plays an important role because it acts as a vector in rDNA technique. Vector means it is a vehicle to carry our desired gene. It acts as a vehicle to carry something. And here our human cell or plant cell which containing our DNA with our desired gene. And in the next step, we have to extract or isolate the plasmid from the E. coli bacillus bacterial cell. This is the plasmid of the bacteria. And we also isolate our DNA from the human cell or plant cell. And next step is we have to separate our desired gene from this DNA. Here we have to isolate this DNA. Here our DNA containing this is the desired gene. Then we have to separate our desired gene from this DNA by electrophoresis method. By electrophoresis method we can separate our desired DNA from desired segment from this DNA. Okay. By electrophoresis we have to separate it. This is our desired gene and this is our plasmid. Then in the next step, by the help of the enzyme restriction endonuclease, cut some fragments in the plasmid, bacterial fragment. And using that same restriction enzyme, we also during the electrophoresis cut our desired gene from this DNA. Okay, for both the cutting we have to use restriction endonuclease enzyme. And next we have to fix our desired DNA into the cutted portion of this plasmid. Here it is fitted, and then it is called as recombinant DNA. This is called our rDNA containing gene X. Here the X means gene of something, means this is our desired gene it is responsible for something pest resistant in plants, or it may be something whatever. Here that gene it contains. Then again this plasmid is inserted into the bacterial cell, and due to cloning there are number of bacterial cell will be formed, and all this bacterial cell contain our desired DNA. This is the step of recombinant DNA.